Okay, so if I are you able, everyone, are you able to see my screen?
Yes.
Yes.
Okay, cool. So if I want to visualize this file, I need to, what I need to do, I'll just clear this by clear, and then I will type head. I just want to see the beginning, and 100 lines I want to see. 2nd December. And all, so this is the name of the file I want to see. So these are the first 100 lines. This was NCBI file that's for my project. So this is how we can see. I can make the screen big, and right now this doesn't make much sense. So if I need to find the specific information, I just need clear, then cut F. I just want to see the 8th, what's in the 8th, and then what's in the 9th column of the file. So the cut command will just fetch me the rows or columns of, not rows, columns of the file. So here, these are the name of the organisms. I can see what's specifically on it. You can do, if you have any tabulated file, if you have that GFF file would work great on it.
We lost your screen. We can't see the.
Wait. Yes, it seems. Next up. Are you able to see now?
Yes.
Yes.
Okay. So this is. Did you download the GFF file? Everyone, anyone?
Yes.
Okay. So here is that GFF file for me. So
Oh, okay.
so I set up my terminal here. So I'm pwd. Now I'm inside my workshop file, okay, and I have my this apis mellifera honeybee GFF file. So I want to see what GFF file really contains. So go to the head, and you can do GCF tab. What next is 3, again tab. It will give you the file. So this is what the GFF file looks like. GFF file means annotation file. So I want to see the first 100 lines because this is so little information. So the 100 and 100. So this is what the GFF file looks like. It will give me the information about the segments of the genome. So this is a gene. This gene is starting from 9,272 position, and it's ending on this. And this gene ID, this is the gene ID, and this is about the gene details of the honeybee. So this is for the honeybee. Are you able to unzip this file as well? If you are able to unzip, you can read this. You can visualize the file by even the GUI like this, but it's going to take a lot of time.
Yeah, it worked.
And For me, VS Code runs automatically. So clear. And so the last line, tail command will give you the last lines. What are the last line? So cat stands for concatenate. So cat, it streams all the information like cat, and the one GFF file I have, I just want to see all the GFF file I have. So it will give me information like this. I can visualize everything together with concatenate. If I have to stop this in between, I just need to press Control and C. So the Control and C command stops. If your program is taking so long and you want to stop it, then you can use Control and C. So this is for the cat. And cat is used to even combine lots of files together. And yeah, cut, I have already talked about the cut. So I just want I just want to see the gene, and then from where it's starting and ending. Cut f, I guess it was on 3 and 4 gc, or I can do whatever the file I have, gff file I have. Control C. So this is giving me 4 and 5. Now this is giving me the only columns, exon, and where it's starting and where it's ending. So this is how we can fetch the information. And one more interesting thing, if you want to save it in another file, the information that you extract, you can do this is a standard output and name it new GFF. This sign will give you output directed file. Like this is just the new GFF I created right now. So that will give me a file. So this is the file I just made here. So this is the file. So the cat command will give it concatenates, and then, and here's important command, that is a grep command. Grep is similar to find and search. So how you find and search, it's similar to that. So I need to do grep, and I want to look stat protein, stat gene. So in all the GFF file I have, so it's giving me about the, it's giving me about this state, not the stat. Stat 5, state is stat. Just stat protein is fine. Control, so this is how the red line is fetching me. So the grep will fetch me the required information, like search in GUI interface. And this symbol is called a pipe. So this pipe is required when you want to merge lot of commands. Like you can merge grep and cut command, and you can merge so many commands together. So for this we do a piping. And now this much is for the basic terminal commands. Now let's talk about NGS workflow. I'll just briefly mention because next generation sequencing data is the most primarily data of like most primary job of a bioinformatics. We are going to handle a lot of NGS data. So library preparation is a wet lab work, and sequencing, there are sequencing centers where we can give our samples for sequencing, and data analysis is our main job being a bioinformatician. So there are a lot of sequencing techniques like chipseq, RNA sequencing particularly is for the gene sequencing, sequencing the where the gene is expressed in a much higher level. So there are a lot of sequencing techniques, but today we are going to talk about only RNA-seq, only the RNA-seq. So these are the various platforms of sequencing. So we have small sequencers like Minions and even the PacBio, and then we have a very big sequence like NovaSeq. These are the platform machines that do sequencing for us, and these are the some bioinformatic approaches. De novo assembly means like knowing the genome for the first time, like if it's completely a different plant that you have discovered. So we have to do a de novo assembly of the genome, and then reference mapping and variant calling for the SNPs, insertion in gene, deletion in gene, as CNV means copy number variants, gene expression, in which conditions gene are expressed, and functional annotations. And the applications we have for like sequencing entire genome, and then long read sequence, exome, targeted sequencing, and transcriptome sequencing, epigenome sequencing to see the methylation, metagenome sequencing when we have a lot of cultures together. When we do environment sampling, we do metagenome sequencing. So this is just an overview of the NGS analysis, NGS platforms. I hope you are clear so far because these are much theoretical things. So RNA-seq data analysis pipeline. I'm talking about only RNA-seq data analysis pipeline, but all the chipseq and all the de novo assembly, they all follow the general pattern. So first there is a processing of a raw data, and then you have to align the reads, and then we have to see the count in the read, and then we have to see the difference in the expression level. So this like doing the QC, quality control, and this aligning, and these all are the same for all sequencing, but I'm just focusing on a RNA-seq here. Only RNA-seq is a different than other. So we do RNA-seq to study the entire transcriptome. So once like DNA will give mRNA will give protein. So we are going to see how the r like how much quantity or what is the difference in the expression level from this RNA-seq. And this RNA-seq also provides information about the alternative splicing and non-coding RNA. So alternative splicing and non-coding RNA are also a similar concept to gene expression, but in alternative splicing we have splicing at a different it's going to give us a lot of isomers or isoforms of mRNA. So this is the this is the first step, is a quality control. I don't know why it's coming like this, but this is the first step for the quality control. So fast QC is a software. So any data we are given from the NGS, we have to do the quality control. Quality control matters in every field, and bioinformatics is no exception. So whatever data we are given, first we have to see if that data is worthy to do the further processing. So this fast QC is the most prominently used software, and it generates HTML QC report. And you can read about the fast Q file format in more depth. uh, I have attached the link here, and this is how how a sequencing file looks like. So we have the name of the sequence, and this DNA sequence, and this is a quality break, and this is the quality scores. So the quality scores are, these are the Fred score. So if we have in this green, so most of here we have a BBBFFF7. So this means we have a good quality of the data. So if we have these ASCII characters, then this data is not suitable for the further downstream processing. I have sent a file. I think you can see the HTML file that I have sent in the mail. So that was a COVID analysis file that I had worked on. So are you able to see my screen for this one was the COVID file that I had, perhaps Delta variant, I guess. Anyone, are you able to see the fast Q report that I have sent?
Yeah, yeah, yeah. We have
Okay,
the HTML.
so I will show you the how raw file looks like. So the NGS machine will give me a raw file like this. So the first will be the name of the identifying name of the sequence, and this is the sequence, COVID sequence, around 100 I have. This is 100, I guess, and this is just a break, and this is the quality. So this is a very good quality sequence. We have very good quality sequence data we had. So I can say it's a very good sequence data, because I have done fast QC. So this is the fast QC report will give me the basic statistics. Here I have to see the QC GC percentage. So GC percentage is a 50, sequence length is a 101, and this is the platform Sanger Illumina. I used Illumina sequencing, and this is the per base sequence quality. You can see that it's very good sequence. This blue color is a median, and most of them are on the green portion. It means this sequence is acceptable. So I will show you how bad sequence also looks like. This is a very bad sequence file. So if somebody gives me this file, I have to really sit and think, no downstreaming process, because this is so bad sequence. You can't really trust if the sequencing machine has worked properly, because this blue line you can see like it's not giving good confidence score. I'm not confident here. At the last portion you can see that the error rate is really high. I can't really say that the last portion the machine is giving me the correct reading. So sequencing plays a very role. Quality control in a sequencing is extremely important, and we don't need this information, but you can read about more about this. I have sent in a link, and this is a sequence quality scores, and this is the GC content, and uh this is the mean number of a sequence, that 101 base pair length sequence I have. And the most important thing that we should look is this one, adapter content. So while sequencing we use lot of adapters like Illumina universal adapter. So in this particular COVID sequence we had used the Nextera transpose sequence. So this is only the adapter. So this from here to here it's a real sequence of coronavirus, but after this it's just an adapter. So we have to, while we do the downstream processing, we have to cut this portion, we have to eliminate this portion. Everybody's clear so far? Hello?
Yes. Yes.
So how you can going to trim the unnecessary portion? So there are softwares, the adapter contamination and low quality reads must be trimmed. So there are cut adapt, triometric. So these are the softwares that we can use, and this is the command line that we have to use. So all the tools that we use for NGS are on the Linux base or a Macs base. So that's why the learning CLI interface, learning the commands in terminal is extremely important, because we have to use these terminal command lines for the downstream processing. So this is the command, so this is the adapter that we had, and we just cut it, and these all are called the flags. So the person, the software developer who made this cut adapt, uh, has already written instructions, so you can read in depth what are these flags stands for, this parameter stands for, in its official website of cut adapt. But this is just for the reference that I have shown, like we can use this command. And this is the mapping. For mapping, so the mapping stands for, we have this small reads, as I have shown you before. In the raw files we have around 100 reads. In that 100 also some are adapters. After removing the adapters there will be around 40 to 60 ATZC reads. So we have to map this in the, for example, if the reference sequence is a Wuhan virus, so we have to match if how much the delta is similar to Wuhan virus. So this mapping comes a role while mapping it with a reference sequence. In this case, because I'm talking about RNA sequencing, so it's mapping with the exons. This blue is exon and this is the intron. So I was talking about the splicing. This is this is a junction between the intron and exon. So there, like it's not always 100% that here will be, sometimes for this mRNA it will be here and it will be here. So those are called alternative splicing. Okay, so there are, the software aligners are there, so some are splice aware, some are splice unaware. So according to the softwares that we are using, we have to map the reads that we get from the NGS. So this is what the mapping means. I don't know why my, okay, maybe I have, I don't know, maybe I shifted this in another, so it's coming very much unclear. Are you able to see my presentation right now? Here it's clear.
Yes.
Yeah, so that because I transported into the another like PPT presentation, that's why it was coming really bad. So slides. Huh. So this is the prompt, this is the command line prompt that we have to use for mapping. So this, I was talking about the splice unaware, splice aware aligners. So these are the type of aligners, and this is the command line. And when we have to compare our reads with the reference of human genome or animal that we are working on, so star is most prominently used one. And it's an spliced aware aligner. So this is just basic about aligner, and these are the flags. You can read about this more when you read about the star on their official website. So post mapping quality check, we have to do quality check even after we do the mapping, because what if we do a mapping but only 20% of our genome is related to Wuhan genome. So that will, like, we can say that okay, it may not be coronavirus, it may be something else. So the we have to do the quality check even after the mapping. And QualiMap is a very popular software to check the quality of the mapping. We have to just run this command on our CLI, and it's a Java application, and we'll get the report in HTML, same like for the fast QC, the initial one. So you can read more in detail about this. And here I have mentioned that if you do the aligning, and if it's aligning is more than 70%, then it's a good aligning, and it will give us the statistics in this way, how much is uniquely aligned and other statistics we can get. And from there we can know that okay, this is a good alignment, and we can publish the paper. So, and this is the final step, differential expression analysis. The goal of RNA-seq is to perform differential analysis of the genes expressed in different conditions, and read count data is obtained from the genes. And differential gene analysis takes normalized read count data and performs statistical analysis to discover qualitative changes in expression level. So these read counts are the counts of mRNAs, and normalization, data normalization is itself a big topic, but once we get the data, we have to do the normalization, and then we have to do the counting. In depth I can send you about normalization on some link, but it's a very big and complicated topic. And for the gene expression, we use a DESeq. It's an RNA package. And if you are confused what does the differential analysis means, briefly I have mentioned this. We have a healthy mouse, and this healthy mouse is expressing this type of genes, but the disease mouse is expressing this kind of genes. So this differences in the expression is called the differential gene analysis that I'm talking about. Okay, and this is the downstream how the data looks like. So these are the name of the genes. Okay, in a healthy, this number of genes are being expressed, and these are not being expressed, and the genes play a role in a ribonucleoprotein complex biogenesis. So these are the downstream process for the RNA analysis, and these are done in R or in Python. So the R and Python programming languages are required for the very downstream analysis, for the data visualization mainly, but mostly we'll be working on command line interfaces. So now this is the finished. Well done everyone. Basic Linux and bulk RNA-seq analysis, data analysis coursework is completed. Anyone wants to tell anything? We have a very short remaining time. Anyone?
It was great, and I want to know more because it's very interesting, and I want to improve and learn it.
Yes, in the beginning of the slide I have I will send the slide to you everyone. And in the beginning of the slide I have mentioned these are the very good online resources you can know further. And Python, for Python, because I may even start a classes or something, because this is the thing that I'm doing since 3 years. So we can more learn on a Python. Yeah, we can stay in touch in a mail, and you can mail me if you have any doubts and queries. We can talk personally over a phone or something, because we are only around 10 participants today. So let's create a kind of a group. We are not strangers, we are colleagues. So let's be in touch, and you can mail me if you have any doubts on a bioinformatics basic stuff. And if you want to join project or something, we can always talk. And the other thing I want to mention is that there is one science outreach servers. It's a supercomputer. So those people who wants to use a supercomputer and they have some ideas on bioinformatics can contact to the scientist. He is Anthony Nash. He's a researcher in Oxford University. So he's providing supercomputers facility to students like us. Rahan, anything you want to say? Let
And
me
what is the use of that? Yeah,
uh,
I don't understand.
so
You said that we can contact him.
yeah, you can contact him if you want to, if you have some project in your mind and you want some
Okay.
super fast computer.
Hmm.
He has, because most of the
Okay.
bioinformatics project or machine learning project or even chemi informatics like simulation run and biophysics thing, it requires a lot of data, and he is a person who can give you access to supercomputers. This is
Hmm.
just for the information, because I use the server, I use the supercomputers available here. That's why I just wanted to share this. Beyond this, anything that I want to say is that thank you so much everyone for participating here. I'm a student like you. I'm a little old student, but I'm a student like you. And I just wanted to share my knowledge I have. And there were so many technical errors in between, but it was fine, I guess. Anything you want to say?
Yeah,
Anyone?
overall it was great. Thank you so much.
Thank you so
Thank
much.
you. Thank you.
Very
Rahan,
Thank you.
I'm not
I
able to
wanted
hear you.
it would be very more helpful for us if you take more sessions on this platform
Thank you.
as a learner and we are learning. And you have mentioned that there are two programming language. One is R and another is Python. So which would you prefer us to learn at first?
Jocelyn, Montasim, Dave, I hope I'm pronouncing right.
Yes, thank you so much, Misha. I don't know if we could create a Slack channel or something to keep in contact so we can share some files
Yes,
or
yes, I will do that. You are on my mail, so I can share the new projects that I am doing, and if I can involve you in that project, because I'm doing such kind of project. So it would be really great to include all of you. And thank you so much everyone. I'll upload this video on a YouTube. I hope you don't mind. And anybody have a problem in setting up Ubuntu or using terminal, just let me know. I will help you personally. No worries. I'm a person like you, so I'll help you no matter what. So for today, this much. We'll be in touch. Okay, goodbye everyone.
thank you.
bye. Thank you so
Bye
much.
Thank
bye.
you. Bye.
Bye.
Bye, thank you.